Hello, thank you for coming to my talk today. My name is Ian Hines. I'm a PhD candidate in Dr. Ann Stevens' lab at Virginia Tech, and I want to give you a talk about one of my projects today, and that is analyzing pathogenic Vibrio parahaemolyticus and oyster interactions in order to prevent human disease. Vibrio parahaemolyticus, or VP for short, is a gram-negative human pathogen and globally is the leading cause of seafood-borne gastroenteritis. The type strain, one of the type strains with which we work is RIM-D. And the number one way in which people become infected with VP is by consuming raw or undercooked shellfish, especially oysters. Oysters are a very important resource to Eastern Virginia, specifically the eastern oysters. And cases of VP infections are still going up worldwide, despite efforts to mitigate infection. And so one area of research that has been relatively understudied has been the interactions between the host and pathogen, oyster and VP. So our lab has hypothesized that VP is capable of actively colonizing oyster tissues versus exhibiting only transient association. In order to study this, we developed the following oyster inoculation system. Starting on day one, we acclimate our oysters overnight within artificial seawater. The next day, we then add those to our aerated tanks, which you can see on the top right of this page. Following aeration or acclimation in those aerated tanks, we then inoculate those tanks on day 3 with our strain of Vibrio. So our strain of Vibrio is VP RIM-D MC. The MC is short for encoding for M cherry fluorescence. It also encodes for chloramphenicol resistance, which is why on day 2 you see these CMs in front of the tanks. The inoculum we use is at about 10 to the 7th CFUs per mil within the tanks. So we allowed the challenge to sit there overnight with these oysters. And then on the next day, we then depurate the oysters. Depuration is where we take oysters from one of these quote-unquote dirty tanks and then add those to one of the fresh seawater tanks that you can kind of see on the left. After 24 hours after depuration, we then harvest our oyster tissues. So these trials run 1 week each, and so the data you're going to see took place over 2 different trials. And the treatments we used for this, you can see at the bottom left. Treatment A was where we had no chloramphenicol or VP. This was our control. Treatment B had chloramphenicol but no VP. In this case, we're just seeing what the native microbiome looks like whenever we challenge it with the chloramphenicol and reduce it. Treatment C is where we added VP but no chloramphenicol, and treatment D is addition of both chloramphenicol and VP. So we first looked at this in the summer this past year, or 2020. And you can see on the left-hand side of this page, this is the summer 2020 graph. We have 2 sets of bar plots here. The first one is TCBS, and this medium is where we select for total Vibrios. So we're not looking at just our strain, but all the Vibrios that are within those oysters at that time. You can see in the blue graph here, the gray and the yellow are all about 10 to the 6th CFUs per gram, whereas the orange bar, this is treatment B that did not have Vibrio, but it did have chloramphenicol. We do see about a 2-log reduction in the total Vibrio microbiome at that point. Looking at the second set of bar plots on the right, you see it says TCBS plus CM10. In this case, it's indicating that this is selecting for just our VP strain. Treatments A and B both recovered 0 CFUs per gram of our strain, so we saw no cross-contamination there. Treatment C, you can see, is about 4 times lower, about 10 to the 5th, or sorry, about 10 times lower, about 10 to the 5th CFUs per gram compared to the total Vibrios that are in those oysters. Treatment D, however, was about 4 times lower than the ones that were found in the total Vibrio system. So we conclude from this that our VP strain is able to colonize these oysters at about 10% when there's no chloramphenicol reduction in the microbiome, and about 25% when we do reduce the microbiome in these oysters. So that native microbiome is clearly important for preventing colonization from external inoculum. We also repeat this experiment in the winter, because we did have some preliminary evidence that showed that the colder months would lead to reduced levels of Vibrios. And indeed, you can see those bar plots on the left-hand side of this graph are about 10 to the 4th CFUs per gram, around that, except for treatment D. Now in treatment D, it's about 10 to the 6th, which is very similar to what we had seen in the summer, which is a little different than the other 3 groups. Looking at just our strain of VP, you see it on the right-hand side of this graph. The treatment C is still making about 10% of the total Vibrio microbiome. However, in treatment D, the total population of Vibrios within this system is our Vibrio strains, the VP-RMD-C. So from this, we can see that not only does the colder weather reduce overall Vibrio loads in the oysters, but it also leads to more efficient colonization by our strain of Vibrio when these oysters are treated with chlorphenicol. So this is going to be really important for us moving forward because our plan is to submit samples for microscopy. We want to make sure that we are getting oysters that have the highest colonization rates by Vibrio. And I'll go into more detail in my future directions. We also know that there are several virulence factors produced by Vibrio parahaemolyticus that do allow for better colonization in its host. And so we also received a couple strains from our collaborator, Dr. Kim Orth at the University of Texas, who has a few of these strains. And so following the same procedure on the right-hand side of the page, we have a couple different treatment groups. So we have our wild-type strain in blue. In orange, we have a mutant of VP-RMD. In this case, it lacks a functional thermostable direct hemolysin, or TDH. It also lacks a functional, with the first functional system of the type 3 secretion system in this Vibrio strain. And our pore 3 mutant, the one in gray, lacks both the TDH as well as the second system of the type 3 secretion system. So following the same procedure that we had in the previous set of slides, we see that we have the TCBS cycling for all Vibrios. On the right-hand side of the page, we have the TCBS plus chlorphenicol. And the important thing here is that all three groups have about 100% colonization efficiency by our strain of VP as compared to the total Vibrio population in these oysters. So we're not seeing that there's a significant reduction in colonization rates by these different mutants. However, if you look on the TCBS medium, and both of them in general, a trend, you can see this trend of pore 3 growing a little bit better in the oysters than the wild type as well as the pore 2 mutants. And so we also looked at the water column in this case as well, and it correlates roughly to the oysters, the proliferation within the oysters, because we see that in the top right graph, you can see that the oysters, the pore 3 mutant on day 2 is about 1 log higher than the other two organisms. And even after depuration, that trend still holds. The pore 3 is about 1 log higher than the wild type and the pore 2 mutant. And so the loss of that type 3 secretion system, number 1, does appear to lead to better survival within the tanks. So we also looked at this in vitro culture as well in the bottom right, just to see if there's any growth rate deficiencies or increases by the pore 3 mutant. You can see that over time, that there is a trend of the pore 3 mutant being able to grow a little bit better than the other two, but by the end of its growth curve, the stationary phase, which is where we're inoculating these oysters, but there is no major difference in growth rates. So that wraps up a couple of projects I'm working on right now. So I mentioned before that we're going to send off samples for microscopy. The reason for this is that we also want to investigate how VP-RMD is localized within oysters. Is it intracellular or does it stay extracellular? Which organs do we see it mostly in? So these questions we want to get answered by immunohistochemistry. We're also going to enumerate the amount of VP within the oysters using qPCR-based method. In this case, you can see this figure at the bottom left. What we'll be doing is we'll be dissecting out these different organs and homogenizing those, and then running those through qPCR with VP-specific primers, just to see which organs have relatively higher loads of the VP. In other words, where is it colonizing the most? This is something that could really help back up the IHC work and the point above. Our last point is that we also want to establish the core eastern oyster microbiome. So our lab does have experience analyzing the microbiomes of other aquatic organisms such as fish, and we found, the one thing is that when you have to chlorphenicol these oysters, that it's knocking down the native microbiome and allowing for better colonization of Vibrio. And so this is something we want to expand upon and investigate to see, what are the constituents of this microbiome. And so with that, I'll wrap up. I'd like to acknowledge Dr. Eric Stapp. He's the one who provided us with the plasmid. Dr. Orth, she's the one working on the IHC work. Oyster sources are in the Chesapeake Bay. Dr. Kuhn's lab is the one helping us out with a lot of the fish husbandry, or the oyster husbandry. And in our lab, Guadalupe, Justin, and Maggie have all helped out with a lot of the plating and a lot of the oyster work in there as well. With that, I will go ahead and wrap up. Thank you.